Good evening, Professor.
Hello. So, are you traveling? I know you said
Uh,
you,
no, I'll be
okay.
traveling tomorrow, but the classes, if I have at IU Luddy, then I am using the campus shuttle, so it's raining, so it might be little
Okay.
chaotic for
All
me to.
right.
Thank you so much for considering and giving online class today.
No, it's okay. I hope everybody else is okay because I didn't see it. You're the only one. Last week you couldn't make it. This week you're the only one here. And so I'm concerned about where you got the email. So if you got it, everyone should have gotten it.
Yeah.
Unless there's a power outage or something like that. There's Hayden. That's right.
Hmm.
Okay, so we're waiting for Dylan and Pedro. Pedro was going to go over some of the homework problems.
Okay.
So because there were some things that we wanted to be sure everybody was comfortable with. So
Okay.
so but I know he's planning on coming because we were until 10 minutes ago we were reviewing what we were going to cover. So I'm first. We'll wait another 3 minutes and then we'll get started.
Okay.
I just want to make sure that in case in case somebody needs help logging in. Hi, Pedro. Okay. Well, I'm sending. Okay, well, let's get started today. Today, we are going to be continuing our work on model replication. I have you scheduled to do some presentations of what you've achieved so far on your projects. I'd like to have people build up as they go forward with their projects. And so I hope you see these presentations and the work is progressive that way. And then I hope if we have time to be able to do a little bit of the phase locking model replication that we started last week and was in the homework a bit. And Pedro wanted to go over some points from the homeworks, the recent homeworks that he thought were worth discussing or clarifying. So again, we'll start with having people present their project status, and then we'll do the homework review, then we'll do the model replication, and then we'll come back to the time series prediction that Pedro had been working on. And if there was by any chance any extra time, we'll do some project brainstorming. So let's start out with student progress project reports. And why don't we start with Varalakshmi? And I'll turn the screen share over to you for your slides.
Is my screen visible, like the presenting screen?
That looks good.
Okay, cool. Yeah. So the topic which I chose was cancer cells response to glucose and lactose concentration. So I have read a bunch of papers and like doing all the um, literature study, and out of which like one paper which was exhibiting mathematical model for the cells in vitro cell culture, and which was undergoing taking all this glucose and lactose, and it was undergoing oxidative and glycolysis pathways. So it was mathematically represented. So I thought like maybe I can convert that module into a tellurium module and try to replicate the results out of it. So before we jump into the model, these are all the preliminary pointers that need to aware so that the model can be understood even better for everyone in this call. So generally, normal cell will undergo oxidative phosphorylation. While doing so, it'll consume lactate, and it will produce ATP, which is an energy for the cell. And this will this is like a normal cell condition. And we have a Warburg effect, which says that uh, for cancer cells, which will not go into oxidative phosphorylation, but rather it will choose anaerobic respiration, which is glycolysis, even though we have like lots of uh, uh, byproducts which is available for oxidative phosphorylation. The reason that cancer cells does this is because it is faster compared to aerobic respiration, and I have plotted a difference between the normal cells and the cancer cells. So the preliminary pathways of normal cells will be oxidative phosphorylation, which uses lactate and like and then produce, we can say, 36 ATP per glucose molecule. And cancer cells will use glycolysis as a preliminary pathways and produce lots and lots of lactate as a byproduct. Like this is one of the characteristics of the cancer cell. So, provided this fact, so there is this mathematical model which explains these two questions. So, what are the dynamics of glucose and lactate uptake while cancer cell is proliferating, and how their concentration varies in time? So, in this model, we are not gonna consider all the dynamics. We are just gonna uh, culture a cancer cell in a petri dish, and we are going to give all the growth factors and everything in a petri dish. So our experiment is controlled in that way, and we are monitoring the proliferation of cells through oxidative phosphorylation as well as glycolysis. And in what cases the cancer cells are going to switch between oxidative phosphorylation and glycolysis, plus uh, what is the uh, uptake of glucose and lactose by these cells. So this is the condition that we are going to look in deeply. And the reason that we mainly choose this study is like to understand the metabolic interaction of a cancer cell, and it can be interpreted in so many different ways. So what we are going to see is a simple metabolic representation of the cancer cell. So this is the mathematical model. So the key parameters that we are mainly focusing here are cell proliferation due to oxidative metabolism, cell proliferation due to glycolysis, and the rate of glucose uptake and rate of lactose uptake. So here, obviously, like if we are breaking down this equation, so the rate of cell proliferation due to oxidative, so we can say increased number of oxidative cell proliferated, and we have two switches here. So this switch chi of L is nothing but a switch which decides that the proliferation should takes place with oxidative metabolism. So it is switching from glycolysis to oxidative. So generally, when it will happen is, uh, so we previously saw that cancer cells usually takes uh, glycolysis, which is anaerobic respiration. So, provided a condition where there is uh, provided a condition where there is no glucose molecule which is available in order to perform anaerobic respiration. So we will have this switch to have the oxidative respiration, and also the counter switch of this, which is chi of L star. Again, it's paired with a switch with glucose, which says like if glucose is going to be lesser than the minimum quantity which is required to perform the glycolysis respiration, then it will switch back to the oxidative proliferation. So the same equation with the complementary is what we are having with cell proliferation with glycolytic respiration. And the glucose uptake and lactose uptake with respect to oxidative and glycolytic respiration. So here we can see with lactate, the lactate is uh, increased with a factor 2 with glycolytic update, because
under each glycolytic pathways, we will get two lactate. So it is actually not consuming and it is introducing two more molecule into the system. So um, apart from this, this switch, which which switches from the glycolytic pathway to oxidative pathway, is also like affected by a parameter called pH and as well as the lactose concentration in the petri dish medium. So yeah, um, having said that, we have two more parameters, which is time constants, which is the time taken for this particular switch to takes place. So basically, tau GO says that the time taken for this particular switch or the frequency of this particular switch from glycolytic pathway to oxidative, and tau OG is like the inversion of whatever that we have seen here. Um, so this is the model, and with the paper, like they have presented like five different cases, which was different papers, which was cultured and given, and these parameters were derived from those papers. So the reason that I took LN18 and LN229 is like the exhibition of these two experiments were compared with each other, and they were looking into what concentration, how they are varying. So I thought like maybe it will be excited, and also like it has like all the parameters mentioned over here. So it was kind of made my task easy over there instead of assuming the confluence and everything. So yeah. Uh, So this is the plot that I have taken to replicate. So this plot generally says like the plot A says like um, with 4 days in a culture medium. So what is the proliferation of glycolytic pathways? And blue and red dotted line says about the cell line 18 to blue and 229 to red, and what is the glucose concentration for all these four cultures. So since it is a glycolytic pathway, glucose is being consumed and lactate is being released. So it is like a typical representation of what we have seen in the model. Yes. So with the experimental results, so I could able to replicate the similar pattern um, of how the proliferation is shown in number of days, and also this is the replication of the glucose concentration and lactose concentration with respect to this. One thing that I wasn't sure about these results are there is no initial concentration of about the medium. So they have mentioned in the paper about the threshold, what could be the maximum threshold of glucose and lactose which can go, and uh, the switching time, but there was no initial concentration. And this value, initial concentration, I just simply assumed from these graphs on the day zero. So I just tried to replicate that. But I could still see the, um, you know, the slight difference in the uh, 299 pattern. So maybe like we should go over. But the parameters were pretty direct. So I could able to get the proper results. So that is what I have noticed from here. And yeah, I'll show the working model.
Sorry, are you there?
Yeah, I'm just trying to share my
So
collab link.
you have to unshare the PowerPoint and then reshare.
Okay. Yes. Can you able to see my screen?
That's fine. Thank you.
Yeah. So yeah. Um, so basically we had two different parameters. So I just uh, initialized two different antimony models for all the parameters and then tried to generate the result. So yeah. So this is the equation that governs with glucose concentration and everything that I have just explained in the PPT. So this is with the lactate concentration, and this is proliferation with respect to oxidative, and this is proliferation with respect to glycolysis analysis. And yeah, the preliminary conditions that I have given here based on the graphs on the day 0. So yeah, and these are the predefined parameters. Um, and yeah, I also individually defined the switches. So after displaying it, so this is the glucose and lactate uh, flow that I've got and the proliferation. So here specifically, we try to suppress the oxidative cell proliferation, and we just seeing the 4 days of the data because if you see the parameter which governs TOG, so we are just doing once in 24 days. So we are just actually seeing the first days of data and the same model and with different um, key constants because these key constants varies with each and every cell line. So this is a different cancer cell line and the particular results. And these are the results that I showed in the PPT.
Uh, what are your next steps, and do you need help with anything?
So I was thinking maybe we can see the parameter significance. So I identified the key parameters which was varying with each cell line was these three. So maybe I can run significance in that line. And I have also, like, again, I have to stop sharing and share my PPT. Okay. So, yeah. So these are some of the things that I was seeing the potential of. Yeah.
So what are you going to do? The sensitivity of what to these parameters?
Towards so basically they have given the confluence of certain parameters based on their environment setup.
No, but
So
what is the output of your simulation?
um, output of the
Yes,
simulation.
what is it that you are going to, when you do a sensitivity analysis, you have to say something is going to change when you change a parameter. What is it that you are going to check for change?
Basically, I'm going to check for like how the switch between the oxidative proliferation and glycolytic proliferation switches with these parameters change
Right,
is
but
something that
numerically, what will that be? What will you measure?
so I'll be measuring PO and PG cell
But
densities.
at what time?
Uh, so for all these given conditions, so each cell line has a different time frame in analyzing that. So it basically depends on. So for according to this paper, there are 5 cell lines that they have identified. So I was planning to just check upon the sensitivity of each parameter with those cell lines with
No, but
defined.
what are you going to measure? What is it that the simulation will give you as a result?
Uh, it gives me PG and PO with respect to time, right?
Right, but so what aspect of PG and PO with respect to time are you going to check for change?
So okay, just give me a minute.
What is this plot? What is the y-axis of this plot?
So it is like number of cells uh
So,
due to glycolysis proliferation.
so is this only PO or PG or both or the
This
sum
is
of them?
only, yeah, this is only PG.
Okay, because that's not labeled, I don't think.
No, this is PG. Yeah, it is not labeled. Mm. So it is PG for 2 different cell lines.
So, and so what is it that you want to check when you change the parameters?
When we change the parameters, so basically the K, G, K, O, and K, L says the consumption of cells due to this particular process, right? So we can see how the rate of this proliferation is varying with different rate constants.
So you want to fit the,
Yeah.
well, I didn't say anything, so tell me what you think is yeah. What is your yeah?
No, no, no. I was telling. So this is a rate. So I want to check how the rate of proliferation is varying with respect to each rate constants.
So how will you measure the rate of proliferation?
So with the slope of this curve, so right now this is with 0 to 4 days. So maybe we can check it with extensive time and also with varying. So here we see the glucose is reducing. So once the glucose goes to null, again it will switch into oxidative proliferation. So how sensitive that each cell line is picking up and how it is varying is what I wanted to check.
So can we go back to the equations for a second? It
Sure.
would be very helpful to have a diagram showing what these equations represent. Because
Okay.
you, not equations, but a diagram. an
Okay.
arrow diagram.
Okay.
And it would also be useful to explain what the chi functions look like. But if I see this equation here, suppose there were no glucose at all, what would the equations look like?
So it will have only oxidative proliferation.
So if there's no glucose at all, you have just the first term here.
Yeah.
Okay. And does the rate of growth depend on the presence of glucose?
Yeah.
No. Where does it depend on the presence of glucose?
So right now, for the example which I have showed, so I have
Stay with the diagram, stay with the equations you have here.
yeah.
Does the rate, suppose there were no glucose, what is the equation?
So if there is no glucose, then we will have oxidative phosphorylation.
Yes.
And we will
So what is the equation that's left when you have no glucose?
so we will have these two terms.
But it isn't the second term zero? Isn't PG zero then?
Yeah. We will have only oxidative phosphorylation.
Right. So what is the
Yeah.
equation? Read it to me.
So 1 by tau. So the rate of the proliferation due to oxidative phosphorylation by the total population. So PO will be the P.
So what will happen? What will happen?
So there will not be any glycolytic.
No, but what will happen to PO?
PO will become okay. So it is 1 by tau into PO. P.
So what happens to the no?
So
What
external.
happens?
Yeah, sorry.
What happens? What is the time course of PO?
1 by tau.
No. Do you remember what a fixed point is? What is a fixed point? What is a steady state? Okay. Don't worry. That's not, I'm not trying to put you on the spot. I think you need to look back at the beginning of the class and the basic concepts of stability and fixed points. So what you have here are what are called logistic equations. And you have logistic equations with interconversion between the two species, which is okay. But you need to understand the behavior of the individual equations first before you can begin to do an analysis of sensitivity. If you changed for any parameter except for P star and TO and TG, tau and TG, would it change the final values of PO and PG?
Which parameter? Like, can you
Any
comment on that?
parameters at all. except for tau O, tau G, and P star.
I guess
Okay.
okay.
Okay. So
I can find that.
okay. So I think we should meet urgently. We need to meet this week. Okay.
Okay.
You've done a lot, but I think that you need to go through this now and understand what the meaning of the terms are. You've done a lot of great work here, but it's going to be important if you want to be able to understand what the significance of these things are, to be able to understand some of the basic concepts of stability, because this is based on simple models of cancer cell proliferation. And if you don't, you need to look back at the base models of logistic growth before you look at the more elaborate models here. Because you can't do things like sensitivity analysis until you decide what things you're going to be what things you're going to be monitoring for change. And so I'm happy to meet with you tomorrow. We'll make an appointment. You'd give me your schedule, and we'll meet tomorrow for an hour and go over this together. Okay?
Yeah. Yes,
And
Professor.
this is fine. I'm glad to see where you are with this. And are there any other comments or suggestions at the moment? Again, I think it's a reasonable model to start with, and now it's important to master the concepts in the model so that you can explain in detail what the meaning of these ideas are and what the mathematics is doing here. So maybe look back in that in a simple mathematical biology textbook at the very basics of how you understand logistic growth and stability for a single variable. And when you understand that, then it'll be easier, I think, to understand this kind of coupled equation that you have here. But something that's rather unusual about this is suppose there were, in reality, what would happen to your cell culture if there were no glucose at all?
They won't grow.
Right. In this equation, what happens?
So yeah, there's oxidative. Okay.
Do they
But
stop growing? Does the growth rate depend on the glucose level?
no.
No. So there's a reason for that, but you need to understand why that is.
Okay.
And looking through it now is not the time to do that. But if you're going to present this material, you have to understand that.
Yeah.
Right. But it's good. That's why it's important to have this kind of check-in so that we can figure out where we need to do basics. There are a couple of minor things I would say. One, I would design your antimony so that you don't have to repeat your code, so that you can load the parameters. So you have a single model where you can load the parameters for any cell type you want. That makes life easier. And also you should annotate the units for every parameter in the code.
Okay.
It's more important to understand the mathematics, but as far as the coding goes, that's also important when you're delivering it. And let's come back to your scroll down to where you showed your slides, where you had done a little bit of simulation.
You mean my collab
You
notebook?
had run some simulate. No, here in the slide deck, you showed your results.
Oh,
Stay
okay.
with the slide deck.
Sorry.
Go to slide, what was it, 9, 10? Okay. Next slide. Okay. Why don't they agree? First of all, do they agree? Maybe is a better question. Do these results agree?
No.
Okay, why not?
Uh, I couldn't see, but the glucose concentration is seeming to go to zero way earlier compared to here.
Did you plot the glucose concentration
Yeah.
in the paper and then in your results?
Yeah. Should I?
Do you have it here to show?
Yeah.
You need them on the same plot though, so you can see what's going on.
Okay, maybe I'll share my desk.
And then we'll move on because we've got to let everybody talk. Again, we're not at the end of the semester, but the point of this is to try to make sure that when we get to the end of the semester, everything works. So don't be nervous if not everything works at this point.
Yeah, this is the plot.
It's not labeled, I don't think. So is this the glucose level on the plot?
Yeah.
Okay, where is the plot from the paper?
Just a minute. Yeah.
So you need to reproduce, scroll back, scroll up.
This
Scroll
one.
up.
This is the okay.
Up. Stop. Show figure 2. Show figure
Okay.
2.
Okay.
Okay. Have you reproduced this plot?
No, I have reproduced figure 3.
But you can't do 3 if you haven't done 2 first.
No, so 2 is with respect to different cell line.
Okay.
So I have chosen these 2 cell lines,
Okay.
LM18
Okay,
and
so
LM20.
I see. So this is glucose level. All
Yeah.
right. Okay. And so this does not reproduce, you're saying?
Yeah.
Okay. So you have to understand then why it's not reproducing, right? So because so we'll have to figure out where there's a mistake in your code or whether maybe they made a mistake. Did they give code for their simulations?
No, it's
No.
just a mathematical
Just the
model.
mathematics. Okay. Great. So tomorrow we'll meet and we'll see if we can figure out maybe they made a mistake. It's certainly possible that they made a mistake either in their parameter values or in their coding. So let's look together and we'll see if we can figure out what's going on. That would be fine. All right. Good.
Yes,
Well, I'm glad. Thank you for being the first to present. And I'm glad you've made progress in terms of building a simulation and getting some results. And now that we know that there's an issue with a specific thing, which is, can you replicate their glucose and lactate results, we can begin to ask the question of what's happening and why it's not doing exactly the same thing. Great. Thank you.
Thank you.
All right, Dylan, let's go to you. Okay, go ahead. Dylan, take it away, please. You're muted, so we can't hear you.
Yep, my bad. All right. Okay, can you guys see this?
You're not in presenter mode, but it's okay. You want to start the slideshow, maybe?
Yeah.
There you go.
Okay.
Perfect. There you go.
Okay, yeah. So I'm doing like a comparison of flex balance analysis models and what the effect of adding constraints are on the like predictive capability of it. And so quick background, flex balance analysis is essentially trying to predict the metabolic network of, in this case, bacteria. And just the like generic model just uses the stoichiometric equations of each of the reactions going on. And then it defines a growth equation based on stoichiometric terms. So like how many lipids, proteins, whatever goes into like the actual growth of a bacteria, they make it a growth equation for that. And then they can use all those equations together to solve for like the fluxes that give that growth. And some of the like, I guess, reasons that this isn't all that good. The first one I didn't include here is that it assumes steady state, which isn't generally like all that right. But I guess just on a population level, they assume that it kind of works that way. But as far as this constraints go, there's no information on the rates of the reactions going on, and there's no information on like the protein, the enzyme like amounts or their characteristics. So like, yeah, their K-cat numbers. And so enzyme constrained FBA addresses those by including some of that information as far as the enzymes go. Okay, so the model that I'm using is called IML1515. It is an E. coli genome scale model. It's the most up-to-date model that they have. And so the model in this form has reactions, metabolite, stoichiometry matrix, which just compares like the inputs of a reaction to the outputs of a reaction, and then genes that encode that each of the enzymes. And for the base model, it has 2,700 reactions, 1,900 metabolites, 1,500 genes, and then bounds, upper and lower bounds of like feasibility for each reaction. This is just the Cobra Pi is like the Python package that kind of runs all of this stuff in the background. And so it creates this matrix and then has some different like linear programming solvers to essentially just run this analysis. And what it does is it creates a stoichiometry matrix, and then it has essentially just the like numbers of your reaction. Negative a negative number in the matrix represents like that's an input, so it's consumed in that reaction, and positive is it's produced. And then one of the other things I'm doing is this FEA, it's flux variability analysis. So this just kind of limits the constraint on the like optimality of the growth. And so it kind of allows you to see, since we don't exactly know like the exact growth equation and all that, this just gives us like how constrained is each reaction in order to like at least have some amount of optimal growth. So for like, I don't know, the glycolysis model, like you probably don't have much lower levels of that, even if you're like not optimal growth or optimal growth. So it just kind of gives you a visualization of like the range that each of these fluxes are predicted to take. And so one of the things I wanted, like, in order to compare these models, I want to have different media conditions. So this is like what the bacteria are grown in, what I guess metabolites they have available to them. And so at the moment, I just have these three ones just to kind of like do a proof of concept, but I'll get later in on of like how I actually want to, what this will actually like be used for. And this just essentially defines, in the model, you have different like uptake reactions as part of that list of 2,700 reactions. So like quick example, if you're feeding it glucose for that uptake reaction, you would just have like negative some number representing that excretion reaction is taking up that glucose across a cell membrane, as an example. Oops. Yeah. So this is just kind of a way that you can define the different conditions that the bacteria is grown in when you're actually comparing it to real world data. And then the first constrained model that I am like doing and comparing it to is the enzyme constrained FBA. And this is the one that I mentioned before. It addresses the kind of unrealistic assumptions on like the enzymes that are in just generic flux balance analysis, which is, it just allows for unlimited enzyme capacity. Any flux that like solves the equation it's allowed for, and it doesn't, it allows for like essentially unlimited amounts of um enzymes as opposed to like there's only a certain number of enzymes and whatever. And so what this ECFBA allows you to do is it kind of addresses that, some of these things where it limits the amount of total protein that can be produced. It includes kind of like ranges for enzyme capacity. And these constraints are just like much more realistic. And so I want to compare like how that affects the, I guess, predictions of it. And so these are the two things that it or the two constraints that it adds. The first one is for each reaction, it adds some like range of, or some information about like actual enzyme rate. And then it also constrains the total like amount of protein that is made in the cell. And one of the like ways I'm going to be comparing this is, since there's so many reactions in like the entire metabolic network is, I don't know, very like hard to untangle and actually like visualize. These are pathways that are labeled in the like IML 1515 model, and will just kind of allow at least a nicer visual comparison for some of these things. So these are just the pathways that are included in the model that allow you to kind of compare the same things without doing all 27,000. And this is just the output of kind of the stuff that you get as like an output of the model once it's solved everything. And what we really care about is this objective value tells us the growth rate of the bacteria. And you can kind of think of it as like a like chemostat, just growth doubling rate of some bacterial population. And then these flux values give you, this flux is for this specific equation, what is the predicted flux through it. And then lower bounds and upper bounds are from the variability analysis. And these are kind of visualizations of those pathways. So the black lines are the optimal like non-flux variability analysis levels of, or like predicted levels, and then the bars are the upper bound and lower bounds from the variability analysis. And I did it for these 3 different conditions of only being fed glucose, acetate, and then wild type is just the model comes like preloaded with certain like I guess metabolites that are included in the model. And then so I just did those for a couple different pathways. And you can see that, I don't know, there's like different, I guess the thing that pops out here is that the different growth conditions affect the actual predictions of all of these individual reactions, which is kind of what we'd expect. And I'll kind of go into later like how I want to actually compare this to like real world data to see how feasible this is. But these are just kind of all of those. And so all of those are just for flux balance analysis. Next steps. So I have the model running and everything for EC FBA. I have it downloaded for thermodynamic flux analysis, which just is a different constraint. But where I ran into an issue is the different models that I have, the like names of everything are different. So when I tried to compare like one reaction in one model to the same reaction in another model, they didn't line up. So I have to somehow figure out how to like directly correlate the reactions in one model to the other model. And they're both based off the IML 1515, but they just come from different papers. So that's kind of where I got stuck this week. But I have the ability to set all of the like growth conditions, run the models under those different conditions, and then compare the like one for one of the different models once the names are actually set up. And so the next kind of things is figure out that naming thing, run the TFA model, which just is a different model. It adds extra constraints. Maybe get to running, there's a EC TFA model, which just compares or like has all of those different constraints. And then I have actual like growth data from like E. coli chemostat stuff, which includes the media conditions it's grown on. So we'll be able to compare each of these models to like actual growth data to see what the improvement in accuracy actually is and like what each of these constraints actually give us. Yeah, so that's these. So compare the different growth rate predictions, the different, and especially like across pathways, the different reaction, I guess, fluxes and what adding the constraints does to each of those fluxes. We can also compare like the variability levels for specific pathways to see if constraints just kind of limit that, and that's what adds to any potential improved, I guess, like growth rate prediction, or if there's something else going on that those constraints do. And then I don't know if I'll get to it, but something you can do is they have like radioisotope labeled carbon. It's fluxomics, but they feed radio labeled carbon into like the growth media, and it allows you to track each carbon molecule through. So it gives you like flux data, but I haven't looked into that too much. So theoretically, that could give you a way of like seeing our individual reactions that are present in that fluxomics data. Do your like predictions match up to it? So that's another thing we could compare, but I haven't gotten that data or anything yet. So that's kind of where I'm at, right? I spent like most of this time this week. The naming convention from the different paper models that I had was not there, and so I have to somehow figure out how to like match up reactions across these models. Or
That's a hard one.
yeah, I think I also might be able to get it to where like in the ECFBA it it has theoretic because it's all based off the same like genome scale metabolic model. It should theoretically have all the same reactions, and all the extra stuff is just the like other constraint equations, because it just the way it works is it just adds those to the matrix. So there might be some way that's easier where I can just cut those out of the model, but those aren't labeled all that nicely either, so it's somewhat of a similar problem. um I just found a paper this morning of like someone doing something similar to this. So they might have these models like already kind of set up that way, but I haven't had a chance to look at if they like did it the same way or any. I don't. There's other tools. I don't know if they're using like I don't know, the same Cobra Pi one that I'm familiar with. So that's kind of where I'm at now. But I think once I get those done, like the actual comparison across all of these things, I don't think should be too bad. And I found some data sets of actual like growth conditions that I can like, I guess, see to compare to real world data.
So one thing that might be helpful for would be to have an example, a simple example from some source, which says, how do you use the outputs of flux balance analysis to actually predict, say, the growth rate in the chemostat? In other words, not with a very complex model, but suppose it says the rate of production of some molecular species is X. How do I use that to actually predict chemostat doubling times? In other words, how do I
Like use one
something just
reaction.
some example to apply some illustration of how to apply the results from flux balance analysis to get a dynamic result like the doubling time?
Are you like, do you mean like get the results from like a single reaction real world data and see if we can constrain the model to that?
No, I'm not asking you for something I think that's trying to be difficult. I'm trying to say, suppose let's look at slide 13 and let's come back to slide 13 for a second. So what are the error bars on slide 13? What do those error bars mean?
Yeah, this is I didn't spend too much time on the graphing. They're not error bars, they are the variability levels. So this black line is the actual like 100% optimal growth rate. And then what you do is you loosen that constraint to like 90%
Okay,
of that
right.
optimal growth rate. And these are like the max and min
Range,
values that
expected range. Okay.
Yeah.
So fine. So suppose that actually everything were exactly on its optimal. So the black line, the black line, the black line, the black line, the black line.
Mhm.
And I had that. What would that tell me about the growth rate of that cell? In other words, suppose that I say, okay, my substrate concentrations are this, and now I calculate what my fluxes are given those substrate concentrations. And now I have all this information. It says, given the particular medium my cell is in, I'm going to get this particular set of fluxes. And now I need to go from those fluxes and say, that says something about how fast the cells are going to grow, right?
Yes.
And so do I just pick one particular molecular species that I know is associated with cell proliferation and say that the growth rate of the cell is proportional to that flux, or is there something else that I do? I'm just trying to understand, how do I take this graph and come up with the growth rate of the cell?
Yeah, so going from this graph to that, I don't know. The way it works now is it has a growth equation with specific metabolites. So if you could get the flux of those metabolites, it might be easier to correlate, or you could do, yeah, I guess you would have to do something of like set one of the values for, say, just this FBA reaction, set it for somewhere in this range, and then solve the equation with that set to that value, and then see what the growth prediction would be.
Well, so I'm actually a little confused. Maybe I wasn't reading these. Uh, um, I'm trying to see what the labels are here, what these.
These are just different reaction, their labels.
Okay. So FBA is an unfortunate name for a reaction if you're doing
Yes.
flux balance enough.
Yeah.
But so these are basically rates of synthesis of some molecular species. Is that right?
Rates of flux
Through a
through a
pathway.
reaction. Yeah, I get, yeah.
Right. But at the end of that pathway, presumably I'm synthesizing something. I'm making ATP, I'm making some lipid, I'm doing something. And so what you're saying is there's some magic formula that says, given this particular set of fluxes, the growth rate of the cell is such and such.
It's essentially, yeah, this is all, it's steady state. So I guess what it is actually saying is that all of the metabolites that go into growth,
Right.
which is stuff like all of that,
Right.
at steady state, all the fluxes of those metabolites get some flux where like all those metabolites are inputs and the output
Right.
is just growth.
Right. And so what I'm suggesting is, can you find a very simple paper, maybe an old paper, which says, this is how we're going to use this information. In other words, my flux balance analysis says my rate of synthesis of ATP is this. My rate of synthesis of inositol triphosphide is this. Therefore, I'm going to say that the growth rate of the cell is this, just to show as an example of how to use these flux rates to estimate the growth of the cell. I mean, presumably the faster the flux, the higher the flux rates are, the faster the cell grows. But if I'm missing some key metabolite, that won't grow. And so I'm just trying to get a sense of how to use this. What you mentioned about being able to actually measure the flux rates using radioactive tracing, of course, that's how people did all of these incredible experiments, getting all of this information about metabolism, was by tracking molecules through these reaction pathways. And so in that case, I mean, in principle, you're actually able to directly measure the flux in a particular pathway. And that is something you can do to validate these results. That's, I suppose, the primary validation for flux balance analysis. And that already is quite interesting. But I'm just trying to understand, if I want to go from this, you said you want to try to estimate the growth rate of particular bacterial species under particular substrate conditions. I just want to try to get a better understanding of how you would use the fluxes you've illustrated in figure 13 to do that.
Yeah, I guess the only way that I'm really familiar with using these is for like knockout experiments for like gene essentiality.
Right.
So I would have to find a paper to see what people use that as far as like
Right.
specific ones.
Right. I mean, that would be fine too, but it wasn't something you mentioned specifically here. And since you mentioned using it to predict growth rate, that's why I was asking the question is it's not clear to me exactly how to do that. Pedro, you said you had some things you wanted to bring up too. Hey, it's a fascinating problem, and I think it's really interesting to see how it works. And I also appreciate your frustration with the fact that you could have basically two versions of the same model that you can't compare because the naming conventions aren't consistent. But
That was a couple hours this weekend.
yeah, but Pedro, go ahead.
Yeah, my issue here is that I mean I'm new to the topic. I've never did flux balance analysis. I don't understand the graph at all. Like I look at the graph, it's meaningless to me. So
Yeah, I
that's the
didn't
problem.
spend as much time as I probably should have on the graphing. But so this is specifically for the glycolysis pathway. These are all the reactions involved, at least that are labeled on that in the model. And so if you just look at the blue ones, that's for the like wild type growth condition, which is just what comes with the model. I didn't actually look into it. So that's saying like there's these certain excretion reactions that are like through the cell membrane. So like if it's excretion glucose, it's how much either glucose is the cell getting rid of or is it coming in. So that's like how you define the different growth conditions. But then what as far as like what these are all the reactions in the glycolysis pathway. And so this is telling you that after you run this equation and solve it when it's at steady state, this black line here is the predicted flux through this equation under this blue condition.
So
The
what
top
is
of it,
flux and flux of what?
the flux is the flux of like if you think about a chemical reaction, I'm gonna butcher this, but like what is it? 6 glucose plus 6 oxygen
Mm-hmm.
goes to 6 water and 6 CO2, whatever that equation is. Like
Okay.
the flux is how much you essentially like use those equation and then have like your feed levels. And so it's how much of the I think it would be how much of like each metabolism level is flowing through this reaction.
Yeah. Is that millimole per gram Dalton watts hour? What is that unit over there?
That's what they gave. Millimole,
I don't understand the units. Okay, so
millimoles
it's
per
whatever
gram.
flux of moles of glucose. So
No, it's when you look at like a specific reaction, it's the flux of the
so you have ENO is the flux of ENO.
ENO is the reaction. That's like yeah, I guess that's actually I guess it would be the flux because it
For each
yeah,
of these
sorry.
reactions, you have one variable that you are measuring the flux of.
Yeah. Now that you say this, I don't know if it's the flux. Maybe it's like the I don't know. I'll have to look that up because now that you say that, I don't know the exact thing it's measuring. It's just
Mhm.
it gives you these like flux values and thinking through it right now. I'm not 100% sure.
Yeah, it's
So
fine. I mean, it's fine. Just in your final presentation, the these kind of answers have to be clear. Otherwise, it's impossible for me to understand what the plots show.
Yeah.
So like, uh, yeah, so like this may not be the only question. So when you present a plot, ask about ask what is the question, what is for each piece of the plot, each part, like these trace, what is this trace, these block, what is this block, this color, what is this color, what is the units, what is in the x-axis. You know, ask this question for each component of the plot, and then maybe put all this information in a caption, and that would be enough, in my opinion. Yeah, so if you provide the information to all these pieces, I would be able to read the caption and understand what the plot shows.
Yeah. That makes sense.
I mean,
Think
this
we-
one you have on the screen now, I'm a little concerned by all the flux values of 0 and pathway, not a number, is just supposed to be the name of the pathway there.
No, that's so only some of the pathways are labeled. So like whatever this reaction is, isn't
Yes.
in one of those 4 pathways. And so I would, since it's a flux of 0, it's probably some like peripheral, not that important in this condition flux or reaction. Or
Okay. I guess then I'm asking the question is what are we supposed to take away from this picture? Coming
this was
back to
just,
Pedro's comment a little bit. In other words, if the fluxes are 0,
yeah, this
I'm
was
not
just,
sure.
I just showed the head of a data frame to show you like kind of this is what you get as an output from it, and like this was just the head of a data frame to kind of show you like what the data from the solver gives you.
Okay, that's fine.
By the way, ChatGPT has obviously been asked the question I specifically asked you.
Did it find a paper?
It's happily generating. No, it's happily generating an estimator. Given the FBA analysis, it's generating a growth estimator for me. I'll share it with you. It may not be right, by the way. I mean, I haven't looked at I haven't walked through this to see whether it's giving me garbage or not. But at least it's trying to explain the question that I asked.
Okay.
And I'm not saying that ChatGPT is a substitute for understanding something, but I do sometimes feel that uh, it's not a bad place to start with. Uh, you can take a look and see whether it says anything useful.
Okay. I'll
Obviously,
also,
I'd rather have you understand it and explain it to me than have ChatGPT do it. But at least it can give me some context for some of the things we're talking.
okay, I'll see if I can find a paper where they do specifically that as well.
Great. All right. Well, we should probably sit down together, and you can try to explain things to me more. You're much more knowledgeable about this than I am in many ways, and so you have to do the educating on your side to help me understand what you're trying to do.
I'll do my best.
Okay.
Put you in
Anything
a bad
else we
spot.
want to talk about today on this? Anyone?
Good.
No. I think we can jump to rowing now. Otherwise, we're going to run out of time. But I mean, yeah, talking about the projects is by far the most important thing because we're heading to the end of the semester. But yeah.
Okay. Royang, do you want to tell us where you are with things? And Dylan, you're going to have to unshare so that he can share.
So the project is very simple. It's just to model the signaling pathway of the MAPK and AKT. And I want to find the drug response to the signaling pathway. And I'm focusing on the PI3K, the inhibition receptor. So I was focusing on the MEK, which is another receptor, but I think it will be more interested in PI3K, so I change it. The overview. So this is the model is from the paper called a computational model on the modulation of this mitogen activated protein kinase, MAPK and AKT pathway. And the paper studies how a ligand called Hergulen activates the ERBB4 receptor and how this signal travels through two major pathways inside the cell. So one is the MAPK to ERK pathway, which controls the cell growth and the differentiation. And the other one is the PI3K to AKT, which controls the survival and they build a large ODE model to understand how the signal moves from the receptor to ERK and AKT over time. And they also use the wet lab data to help them build this ODE model. So here's the model. So it's a deterministic ODE model, which has 34 reactions and 40 chemical, 40 molecular species. And here's the model, and the HRG, which is the input signal, it will bind with the receptor, the ERBB4, which is not shown here. And eventually it will become the RP, and it will activate two pathway. And there's two pathway, and one branch is the ERK branch on the left side, and the other branch is the AKT one on the right side. And it's simple model comparing to their model. And they have many parameters, and I didn't change them. And they also have all the equations shown, and they build the model in SBML. And the reaction 19 is the ERK I was focusing on. Maybe I will focusing on the ERK later because it's a very common receptor. ERK is the the one that control the cell growth. So maybe I will try to focus on it later. So I'm currently focusing on the AKT, which control the cell survival, and the one I'm looking at, the PI3K is a common receptor for drugs. So there's a lot of cancer drug that target the PI3K, so which is a very good target to monitor, to model. And the two major pathway is the SHC to RS to RF to MEK to ERK, which is the left one, and PI3K to AKT, which is the right one. There is a cross here, and the AKT will inhibit RAF, which will reduce the amount of ERK. And so I reproduced the model. So they already provide a SBML code, and I used the tool that converted to antimony and do some reproduction first. And they monitor the ERK double phosphorylation and the AKT double phosphorylation, which is controlling the cell growth and cell survival. And the first plot is how the input signal will affect all the important components. And basically it's just controlling the input amount of HRG here. So it's very simple to replicate. And another interesting plot of this paper is that they are trying to simulate a drug response as well. So they use this wartamine, which is a fungal metabolite, which will target the PI3K inhibitor. And they try different inhibition level of PI3K. So they directly change the parameters. So the reaction rate, they directly change the reaction rate to 10% and 1%, and they will have this. So PIK3 will inhibit PI3K will directly affect the AKT double phosphorylation amount, because without PI3K, it will block the pathway. And also it will affect the ERK because AKT will inhibit ERK. So as we can see here, the more inhibition, the higher the ERK level. So, but this is too simple because they just changed the parameter here. So I've been thinking about adding a two reaction that will bind the PI3K and see if we can simulate the drug response like in real life. So I add two. So I search a drug. I search in the drug database to find a drug that inhibit the PI3K. So I found this one, which is a very common drug targeting the PI3K. And I Googled it, and how it works is that it will bind with the PI3K so that it will reduce the amount of PI3K in the cell. So I add two equation, but here's the confusing part. So the author add PI3K star, which isn't a real protein in reality. It's just an active form of PI3K. We can see here. So the PI3K in this model won't affect the AKT production directly because only PI3K star will control the reaction from PI to PIP3.
Is the starred form the phosphorylated form?
It's just an active form. I don't think it's a phosphorylation form. I'm not sure. Because if it's phosphorylation, it should be PI3KP, right? I'm not entirely sure. But it will be easier if we assume that the drug is targeting the PI3K star directly. And I tried to target this one before, and I don't know how to make it work. So I instead target the PI3K star, which is the active form of PI3K. So it should be the same. So I add two equation to this because the PI3K star will affect the reaction from PI to PIP3. So it will have a direct interaction with this reaction. And I add two equation, which is the PI3K star with the drug that is binding a drug, and another reaction, which is it reverse to this. And I add two parameters, which is the reaction rate from the PI3K star to the binding rate and the unbinding rate. And so in the drug database, I can find that the geometric mean of the IC value is 3.36 and here we are close. So the 50% rate is around 3.15, and
So this is a dose response curve here then.
I think so.
So you're estimating the P50 for it. Okay. So basically 2 micromolar, no, 20 micromolar. So no, 2 micromolar, which is a lot, but okay. All right. Sorry, go ahead. I looked up, since I didn't know the difference between the activated and inactivated form. According to Google, at least the activated form of PI3 kinase is it's a conformational transformation of the molecule and also a translocation from the cytosol to the membrane. Not that it matters from the point of view of your simulation, but it's always nice to know what's happening there.
It's very confusing because I don't think if I change this directly, so it will have to go through this direction. So it will be easier to to treat these PI3K stars the receptor. It will be much easier.
Right.
Not go through all this.
Well, I suppose you could, if you wanted to, you could start lower down in your network and then work your way up. But it looks like it's important to have that. It looks like it's a pretty fundamental question in this case. I'm sorry, I didn't mean to interrupt you. I was just interested since I didn't know the difference. A lot of times that star is used for phosphorylation, but it's not in this case.
Yeah. So I would maybe try to treat this one as the receptor later to see how it go through this pathway. And it will be more interesting. But this one is more directly affecting this, so it will be easier to tuning the parameters. So I need to tune the reaction rate to match the drug response, which is the IC50 value. So IC50 value should be the value that with this concentration of the drug, it will have like 50% of the cell die. That's what I assume. So maybe 50% of the AKT value which control the cell survival will equal to this. But I don't know how to.
Yeah, it's a little hard to relate the level to the level of the molecule to the survival of the cell. I mean, I suppose as a zeroth order guess, you could just say you'll assume that the IC50 is when the concentration is half of what it was before. That's probably not right, but it's not a terrible place to start with your assumption.
Because it's not LC50. LC50 is the lethal. So this one IC50 is just the, I think by the definition of IC50, it's not the cell dies. It's just half.
Yeah, so in that case, that's exactly what you've got. So if that curve is the IC50, well, the plotted curve was the IC50, the experimental curve was the IC50, or the go back to the experimental data set that you showed from the drug manufacturer.
The IC50.
So that's IC50. So then it really should be 10 micromolar, more or less, and let's go back to your plot, your graph. So in yours, it's about 2 micromolar. Well, that's not that far off. Doesn't seem to be terrible.
But the mean value is close.
Yeah, so it seems to me that you're actually doing pretty well in reproducing that. Yeah. So what do you want? That's impressive. What do you want to do next with this? How are you going to move forward with this?
Because I was targeting PI3K star, which
Right.
is
Right.
maybe I need to change the target to PI3K because I didn't make it work at first because it goes through all this pathway to go.
Those extra loops.
Yeah.
But you said that they had their published model had all of this in it?
Yeah,
Was
they have every reaction in this picture.
Okay. And so when you run the published model, are you able to reproduce the figures in the original paper?
What figure?
I don't know. I mean, the model came from somewhere, right?
You mean the figure in paper? This is all the figures in papers.
And you're able to reproduce those.
Yeah, it's exactly the same, I think.
Okay. So the model reproduces or replicates. And so then the question is, how do you apply it? So the issue is basically to understand how to apply this simulation.
Yeah.
Is that right?
Yeah. So the problem will be how to adjust the parameter to have the similar plot like this one.
Right.
We change the target to the PI3K. directly.
Well, so I suppose you could you could do knockout experiments.
Yeah.
Of course, you knock something out in the middle of this, it's going to kill the whole thing off. But you could do a knockdown experiment or change. You could certainly do an equivalent where you change each, all of the major rate constants or Michaelis constants and see what happens to the output. That's possible. So the kind of sensitivity analysis that Lakshmi was proposing, I think you could do pretty well. And so your goal is to really to see what. Let's come back to your initial diagram. So what you wanted was, as a readout, was the amount of MAP kinase of ERK. Well, again, come back to, let's just say, I know you said it, but please say it again. So your concern is the amount of what ultimately? If you could do anything, what would the amount, what would the output, what would your answer be?
So originally I was focusing on ERK, but ERK
Right.
control the cell growth, so it doesn't match the GDSC data, which is I don't know if it's cell growth or cell survival of this, so I'm not sure. So the original plan was to match the drug response data and the
Right.
knockout data to the
Right.
model. And
But I think okay, go ahead.
I don't have any more thought.
But I mean, but suppose that you step back from the ability to get the experimental data set for the moment. So let's come back to your diagram for a minute.
Do you start with?
And so now you say the amount of phosphorylated ERK is going to somehow tell you something about the growth rate of the cell, for example. And so now you could ask the question, if I change any of the rate laws within this by, say, if I had a drug that bound to PI3 kinase or to one of these other molecular species to sequester it, to slow down one of these pathways, what would happen to the ERK2P level, for example? Now, you may not have experimental data for that, but it could tell you, if I were going to target some part of this system, which of the molecular species would be, or which one of these reactions would be the one that would be most have the biggest effect on the ERKP2P level. So that would be possible.
Yeah, we
So,
can. So there's knockout, right? The knockout
right.
data in. So we can find some knockout data and
Right.
can rank them. So
Right.
have a list and we can rank them by
Right.
the gene expression
Right.
data after the
Right. Right.
and we can rank them and we will see changing the parameter in this model and how it will affect
Right.
the ERK
Right. Right.
TP and
Yeah.
and we rank them and we see the two rank if it's
Right.
if it tells the same story.
Right. I think that's reasonable. You don't, I mean, it would be nice to have experimental data, but even if you don't, I think you could still say what pathways. I mean, the one thing I would say is, for a lot of these things, I mean, if you do a full knockout, I mean, you can design a knockout that's going to kill if you knock out something down at the bottom, you won't have any RK2P, for example. You won't have any of it. And so you might, instead of doing a full knockout, you might say, if I change the rate, the level or the rate of anything by 10%, what happens to the ERK2P? And so do a sensitivity analysis where I say, okay, I change a little bit like what you did, well, in the diagram you just did here, right? You change the concentration of, well, scroll down to your sensitivity that you just did. Scroll down. Right, this plot here where you have the concentration of your drug and then you have the peak activation. So you could do that essentially. You can create a fictional drug that affects each one of the major pathways and ask the question here, maybe not in micromolar concentration, but if I knock it down by 10%, what happens? If I knock it down by 20%, what happens? And so I could repeat this not as a direct molar concentration, but as a function of the activation or inhibition or sequestration of any particular species. I mean, the reason I'm not wanting to use the word knockout is if I talk about knockout, in principle, I'm eliminating something completely.
Right.
And no drug or almost no drug ever actually eliminates something 100%. It usually reduces the amount.
So they have
Right.
done this before. They try
Right.
to reduce the amount of PI3K to
Right.
1% and 1% and see
There.
how
Right.
it will affect the ERK.
Right. Right. So that's the right-hand plot there, right?
Yeah, this is the
Yeah.
NK.
Okay, great. Perfect. So yeah, so I think you could do a study of that kind for other species or other rate constants in the diagram. And I think that could be quite interesting to see. And then you could say the ones that have the most sensitivity are the ones that you would target. So that makes a lot of sense to me. I'm not saying that's the only thing you could do, but I think it's possible to do that.
Yeah.
Okay.
Yeah.
All right. Pedro, did you have any suggestions, or Hayden, anybody else? I think this is, again, we should maybe try to meet and talk about it a little bit more, but I think this is quite interesting to see how one might go about doing this.
I don't have comments here. I think it's looking good. A lot of progress.
Yeah, no questions for me either.
Okay, so let's try to meet. I keep saying let's meet, and then we don't wind up doing it. It's really important at the end of the semester that I get to talk to you one-on-one so that we can have some more time to think about things. But I appreciate everybody's work, and I realize that when you pick the projects on your own, it can be challenging. And so I do appreciate it. All right, so let's come back to Pedro. Let me grab the screen share for a second. Where's my screen? All right. So what happened to my right. All right. Thank you, everyone. That was great. Now I'd like to turn the floor over to Pedro a little bit. Pedro assigned a couple of problems in the homeworks, and people grappled valiantly with those problems, but I think it would be it's more beneficial to go over the possible answers and discuss them together, so that you can see what made sense, what didn't make sense. You can ask questions about what you understood and what didn't, so that we make sure we can consolidate some of the things that were covered in the homeworks. So I'll turn the floor over to Pedro. For example, there was this problem where you were going to generate a predictor of a quadratic equation, and the basic idea was to do something fairly simple where you have 3 parameters plus the x value, and you're going to predict the y value. So you have a network that starts with 4 inputs and gives 1 output, potentially with quite a few nodes in the middle layer. And several people understood this to be you were going to have 103 inputs and 100 outputs, and then the models didn't train and didn't predict. And that's something that's interesting. There are also some questions about what made sense when you were analyzing data for statistics that Pedro wanted to go over. And then there was a second homework problem where you were going to use a long-term, short-term predictor to do some things. And we played with that in class last time, but in the homework, people had a little bit of trouble getting that to work. And so Pedro wanted to go over some of those ideas with you to make sure that everybody was able to really master that material. And so I'll turn the floor over to Pedro to discuss these couple of problems.
Yeah, just are you guys hearing me well? All right. Okay. So that there are visitors in the house, so I reduced the gain and got the mic very close here. So if it's too noisy, let me know. All right, so for the parabola predictor, we have the new LSTM, the LSTM basics that we used for the surrogate. That's the correct architecture. So you have the three input parameters. You run that over one or two linear layers, and then you pass that through an LSTM that could be one or two stacked LSTMs, and then you have your time series output. That would be the correct approach, and that leads to correct extrapolation of the parabola. To be screen
Sorry,
sharing,
Pedro, were
yes.
you supposed to be screen sharing?
Yes, I'm screen sharing my PowerPoint.
Okay. Sorry about that.
Yeah. Okay.
That's me. That was my Zoom was set in a funny way. Thank you. Sorry about that.
No problem. Okay, is everybody seeing my...
I see, yeah.
Okay. So, but before we touch on that, let's prioritize the questions, the homework questions. So these are case scenarios where you have to analyze what's going on and come up with a solution for the problem or a diagnosis of the problem. So that's the idea behind these these set of questions, and they involve the chi-square and fitting problems that we covered 3 weeks ago. And we are going to go over the questions one by one to make sure we understand these concepts. So question A, you fit a model to experimental data and plot the residuals. The histogram of the residuals is flat, homogeneous. What are the potential underlying issues and what would you do to fix the problem? So Dylan, can you
fit
do you have any idea on this one? So you have your
It
residuals here,
has, if the residuals are flat,
and they're like this. They're distributed like this.
your
So
bias term is off.
this is your histogram of residuals, and it's something
Yeah.
flat. Come again?
Like you don't have a bias term, so it's just offset by some amount. I forget for this exact equation.
So when you fit something, you have your experimental points here.
Yeah.
And when you fit something, so let's suppose your curve does this, okay?
Mhm.
You have a deviation from your fit to the point. And these deviations are thought to be normally distributed if
Yeah.
your fit corresponds to the observed experimental data, right? So if the
Mhm.
fit corresponds, your residuals are normally distributed. So if you plot the histogram of the residuals and they are flat, it means that they are not normally distributed. Therefore, your fit does not correspond to the experimental data. So you're expecting something like this, something normally distributed, but you get something flat.
So that just is telling you you have the wrong model or something.
It tells you that your model does not fit the data appropriately.
Yeah.
So imagine you have so instead of this curve over here, you have something that is a flat line, and then the flat line falls over here. Chances are your histogram of residuals are not going to be a Gaussian. It's going to be something else. Or imagine like let me give like a very drastic example where you would actually get this result. Let's suppose you have like a hill function data. So you have a few points here, and then it climbs up, and then you have more points there. If you're fitting a horizontal line like this, your histogram of residuals will be pretty much homogeneous, right? Because you have these guys here and these guys here. These differences are pretty much the same, so we'll get something homogeneous.
Yeah. Okay. That makes sense. I was confused because if your residuals are perfectly flat, I was taking that as like it means you're almost fitting it perfectly, just offset to some degree was what I was thinking with it. But
Oh,
that makes
yeah,
sense how that could
yeah.
happen.
Well, that like a perfect fit would be that your residuals or the values of the residuals are very low.
Yeah, but I guess like perfectly fit as far as shape, but offset to some degree is yeah. Okay. This makes sense though if it's just a line through that.
If they are offset by us by some degree, and I don't think I did the correct example here. So suppose you have your points here and your fit falls like here, and you
Mhm.
have all the residuals the same, right? Like this.
Mhm.
You'll have like 0, 0, 0, and then a spike around a certain residual value, which is the same as this, right? And in the
Oh.
other case I showed, where you have something like this, it would be a double spike. Yeah,
Yeah, okay. I was thinking residuals were on the y-axis. Okay.
like this. Yeah. So you expect something that is that follows a Gaussian.
Hmm.
This is what you expect. So if you offset like this and around zero, of course, around zero. So if you offset like this, you get a spike. But even if you offset and there is like kind of a Gaussian distribution around this bias, you get a Gaussian distribution that is not around zero, which means you have bias, therefore the fit didn't work. All right? Is that clear now?
Yeah, yeah, that's clear.
All right. So let's go to B and I'll read it together and then Varalakshmi will answer to us for us. A model with 4 parameters fits 50 data points. The reduced chi-squared stochastic. Oh, I think I cut this short. Hmm. Let's go to C and then I will check for B later. Because chi-squared stochastic doesn't make sense. So probably I got the wrong question there. Homework. That's homework 7.
I'll look it up while you're doing the next one. No,
Oh,
you got it.
Statistics. Oh, okay. Oh, I read stochastic. Oh my God. Okay, fine. The reduced chi-squared statistics comes out to chi-squared equals 12.5. What is going on with the fit? That this number is so large, and what are the potential issues with the model or the data? So, Varalakshmi, do you have any idea on this one?
Okay. Chi-square value is 12.5, which is so uh, large. So maybe it has like more deviation with the original data points. And the parameters are 4 and the total number of data points we have seen 50. So it looks proper because generally we consider to use less number of parameters and more data points to see the fit. So I was confused here why we have larger value unless until the model was overfit by the value. But
So if the chi-squared is very large, it means that the residuals are very large.
mhm,
So the model doesn't properly fit the data.
yeah,
That's what it means. The reduced chi-squared, if it is very large, the residuals are very large. And the residuals, again, are the differences between the fitting curve and the data. So what is going wrong with the fit? The fit doesn't match the data points. Doesn't pass through the data points
yeah.
close enough. So I guess an example of this would be, so you have a lot of data points in that example. And then you fit something like that completely does not correspond to the data. Then you'll have larger residuals, and those will lead to a large reduced chi-squared. So it's poor fitting quality, large chi-squared statistics. Okay. Any questions here?
Yeah, that's clear.
Or another example would be, so there are two potential problems. So one is you are misfitting. So you have your points like this, and then you are using the wrong model. But it could also be that you're using the right model, but your experimental points, they don't make any sense. They have like huge error bars, and they are all scattered, and then you're trying to fit something through these points, but the data is so bad that your chi-squared is like your residuals are over the roof, and then your chi-squared will be very very large.
There was
Your
also
reduced chi-squared. Hmm?
something about like when you're recording your data, you like underestimate the uncertainty or something like that, right?
That's another case.
Yeah.
That's another case. But yeah, not here. Not for large chi-squared statistics.
Okay.
Large reduced chi-squared means you're either misfitting the data or the data is too noisy, too all over the place. All right, C. That's for rowing then. You calculate chi-squared, reduced chi-squared equals 0.3 for a model fit. What is going on with the fit that this number is so low? Is it a problem with the data or a potential problem with the model, and why?
It could be overfitting, and also it could be the degree of freedom too large, so that the reduced chi-square is so small. Um,
But like if you have a very large degree of freedom, it doesn't bring the reduced chi-squared below 1 because you also have more residuals that you're summing over. So if we like the equation is 1 over the degrees of freedom, but you are summing this over all the residuals normalized by the standard deviation. So if you have more degrees of freedom, you have more residuals, normalize the residuals, so they compensate each other. So this number in the end, if you have a good fit and very large degrees of freedom, this number is 1. It doesn't go below 1. So your answer is incorrect.
So it's just overfitting.
Overfitting would lead to a reduced chi-squared larger than 1 because you have lower degrees of freedom. So that fraction will increase the value of the reduced chi-squared. So it can't be overfitting. Right? You have 1 over the degrees of freedom. If you are overfitting, your degrees of freedom are very, very low. This is what happens in an overfit. So and if your degrees of freedom are very low, this fraction goes very high. Therefore, your reduced chi-squared goes very large. And this is not the answer for B because we have 4 parameters and 50 data points, so it can't be overfitting for question B. It cannot be overfitting for question C because the reduced chi-squared is 0.3. So does anybody else have an answer for question C?
Your standard deviation would have to be really high.
Correct. Higher than the expected deviation shown by the residuals.
Yeah.
There is a neat way of saying this, which is in the slides. And you just talked about this when we were discussing the question B.
Yeah, you overestimate your uncertainty, underestimate your
Precisely.
uncertainty.
You're
Which one?
overestimating the uncertainties of the experimental data.
Yeah.
Right. That's right. So the only way, again, our equation for the reduced chi-squared is 1 over the degrees of freedom summed summing the residuals divided by the standard deviation. The only way this can go below 1 is if your sigma is very large. So you're overestimating your experimental uncertainties. Okay? Because if you are not overestimating your uncertainties, on average, this fraction here is 1. And then this here on average is n, and then n divided by df is greater or equal than 1. Right? So if I go back to my slides, let me see if I can find that goodness of fit here. Do you see the goodness of fit slide deck here?
Yeah.
Or okay. Oh, this is about the histogram of residuals has to look like a Gaussian, so the other one was looking like a flat thing. So here the reduced chi-squared, we have 1 over n minus p, and here are our residuals. This is our standard deviation. Oh yeah, they are squared. I forgot about that. And then here we have what happens to chi-squared when the model is a good fit. We have that the closest to 1, and this approximates the value of chi-squared approximates to n over n minus p. So chi-squared approximately 1 is the best possible. If it's much greater than 1, deviations between model predictions and data is larger than expected from experimental uncertainties, therefore the model is inadequate. If it's lower than 0, residuals smaller than expected suggest overestimated uncertainties right here. And yeah, so those are the answers. All right, let's go to D now with Dylan. Back with Dylan. You have a system with two variables, A and B. However, the experimentalist gives you good data about A only. In what conditions is B also identifiable? Any idea here?
Yeah, you'd have to have like A tied to B. You'd have to have some equation with A and B, like interchanging them.
Yeah, correct. So if you have a feedback loop, positive or negative feedback loop between A and B, which means information of a variable A provides information about variable B, the parameters of both equations can be identified. Okay. Any questions for case D? All right. So next one. A student fits a model with 12 parameters to 20 data points and reports chi-squared approximately 1. Is this a good result? Why? Varalakshmi, do you have a question here?
Mm, Reduced chi-square value is one, which means it is a good result. But I would have a contradictory opinion because the data point is only 20, which is a very less data point. So
So you would expect a larger reduced chi-squared, right?
exactly, yeah.
All right. Yeah, perfect. That's the answer. Correct. Any questions here from the other students? So this is probably not a good fit, even though the reduced chi-squared is approximately 1. Why? Because we have low degrees of freedom. All right, next, for rowing. Consider the model A goes to nothing with with rate A times K1 over K2. Are K1 and K2 identifiable? How would their likelihood profiles look like?
I don't think the K1 and K2 can be identifiable separately, but the ratio is identifiable. And the profile will be flat along the ratio.
Around the ratio, the likelihood profile will be flat, period. Okay? Around the ratio doesn't make sense, would be around the optimal fit result. Right? Because the likelihood you vary your variable or your parameter around the result of the fit of the optimization, right? So around the ratio, if you just cut around the ratio and you say their likelihood profiles will be flat, perfect answer. Any questions here? All right, G for Dylan. In a likelihood profile analysis, parameter P1 shows a sharp U-shaped curve crossing the cutoff on both sides, while parameter P2 shows a flat curve that never crosses the cutoff. Which parameter is identifiable and what does the flat profile of P2 imply? Which parameter is the model more sensitive to?
P1 is identifiable.
Mm-hmm.
What does the flat profile? The P2 is not identifiable,
Mm-hmm.
and it's more sensitive to P1 because
Mm-hmm.
yeah, it has to be like in that range.
So what would the non-identifiability of P2 imply in terms of the fit?
That you could vary its value to anything and still fit the data. Like it's not tied to the like data, or it's like offset by all the other values or something, or it's like because that's saying if you change the value of P2, it doesn't change the fitness.
Yes, doesn't change the fit. So does that tell you something about a problem with the fitting procedure?
With the fitting procedure.
Yeah, or the data or the experiment.
No, just uh, I don't know that one. With the, probably the fitting procedure that you don't need that variable where you have like it doesn't you don't have enough data to like
Correct. That's the
train all
point.
these. Yeah, yeah.
The data you have does not provide you information about the specific parameter.
Yeah.
Correct. H, you are asked to advise a lab on which variables to measure. Measuring X is expensive, but it allows the identification of two additional parameters. Measuring Y is cheap, but it adds no new identifiability. Which measurement would you recommend and why? Varalakshmi.
I would recommend measuring X because there is no use if a new parameter is not going to give us a new identifiability.
Yeah, correct. That's what I would say as well. Yeah. Unless you measure Y just to show that there is no new identifiability to advise other labs to not do measurement of Y. Right. I, for rowing, a student reports that their optimization routine gives different parameter estimates depending on their initial guesses. What is the likely issue and what strategies could be used to address it?
It could be starting at the local minimum.
Mm-hmm.
And one way is to start with higher learning rate and then lower it after a few epoch.
We are not talking about neural networks optimization, training. We're talking about fitting,
Oh,
right?
like use different fitting method, like the
Yeah.
differentiate, the one we use the Evo differentiate. I forgot the name.
The differential
Evolution.
evolution.
Yeah, that one.
Yeah. So genetic algorithms is one possibility. The other possibility is annealing, where it's more or less what we're talking about the first one, which is you systematically increase and decrease the learning rate, the step size of the optimizer to skip on these local minima. Right, good. Next one, J for Dylan. A model of metabolism has been fit to experimental data with reduced chi-squared close to 1 with 1,000 data points. The modelers report that all parameters are identifiable with the respective values: age, height, weight, exercise intensity, and heart rate. Should you be skeptical of the results? Why? And I forgot to, of course, I forgot to put units. So this is BPM, beats per minute. Go ahead.
Yeah. Yes, skeptical, because like that's a categorical, so it isn't clear exactly how that's represented. And then I don't I think 180 resting heart rate is really high.
Yeah, right.
I,
Yeah, that's the
I'm
problem.
not sure what it's supposed to be, but that feels really high.
Yeah, that's the problem. So even though the model fits and the reduced chi-squared is very good and all parameters are identifiable, which is the best scenario, you are estimating a parameter outside the the biologically accepted range of rest heart rate. Of course, maybe more detail is needed. Maybe this person rest heart rate is 180 because of a certain condition, but that's not informed. Uh, However, there is a reason to be skeptical, depending on the context here, because of the elevated rest heart rate. So this is something that always worries us, which is sometimes your parameter aggregates other phenomena, and then you are overestimating or underestimating your parameters, even though your model fits very, very well. So this is a nightmare that haunts all modelers, especially in biology, because parameters often aggregate a bunch of um, processes. Is that clear for everyone? All right, so K for Varalakshmi. What is the value of the chi-squared that corresponds to a confidence level of 95%, considering a fit with 100 data points and 5 parameters? Explain how you calculated it. Varalakshmi,
Okay.
please.
I have first calculated the degree of freedom over here, which is number of data points minus the number of parameters. So here with this problem, it's 95. And I just looked into a chi-square value with a confidence interval of 0.95 at the degrees of freedom, 95. Yeah. So it's been so long since I have used chi-square table, so I just calculated with Python and then filled it in.
Okay, all right, yeah. So basically you go to the chi-square distribution with 95 degrees of freedom, and then you get the chi-squared value that gives you a 95% area under the curve. Okay, 95% area under the curve. So you have your chi-square distribution for 95, whatever it is, something like this. And then you go to 95% area under the curve integral of the distribution. And this is your value of reduced chi-squared right here. All right. So often, how do you define if your chi-squared is enough? You define a confidence level and go from there. And I bet I drew the thing wrong. So I'm sorry for that. Last one for rowing. The chi-square distribution was not what's that? So okay, this is like this is not a question. This is a item of question K like this. All right, so that's all. I planned to do exercises, further exercises on the surrogate, including GPU. So if we had time, we would introduce GPU usage to our surrogate. Then we would implement the SIR model. So we did like for a dummy model 1 week ago. So we would substitute that by the actual SIR model with loss of immune resistance. Then we're going to introduce new variables as initial conditions and change the structure and change the code to allow more variables as initial condition. And then finally add another observable. So change the model so that the SIR model has two observables, and those would be the vaccinated population and the infected population, because those are two quantities that the government usually keeps track of. And then the surrogate will then make a prediction of two variables instead of just one. Unfortunately, we don't have time to do that today. So I will leave you guys with, I'll leave you guys right here. So if you have any questions about the homework, feel free to ask me now.
You guys have the answers for the LS or for I don't know which
For these ones?
for like the last homework where we were doing the surrogate model stuff because it was not working at all for me. And I was just wondering if you have like solutions.
Which one?
I think it's 8, the neural network one.
For the parabola?
Yeah.
All right. So the issue with that one is so let me show you something that I have from another student here. The idea here was to start from the LSTM code we had for the sine wave, which I saw you did. So this is the structure we had. We have LSTM cell stacked with LSTM cell. And then we had a scan over past data and a scan over future data for prediction. And then in class, we run this for sine waves. So if you run for like 5 more steps, it predicts correctly here. So the idea was to extend that to parabola. So if you just change from the sine wave to the parabola here in your data generation for parabola, so you have y equals this, and then you have a bunch of parabolas here doing this with the exact same structure. If you train this, you will get a correct prediction for the parabola like this. Okay? So if you just substitute one by the other, keeping the same structure. So now the challenge here is instead of providing past data, you provide the parameters. So instead of having a loop over past data like this, you will just have a linear layer that goes from the 3 parabola parameters to the initial ht1. And so then you don't need this. You will always be predicting. So you only have future. The issue is if you do that, you get numerical instability, which is what I think you got. So basically, the issue is that this LSTM cell is inappropriate. It leads to numerical instability, as far as I've looked into it. So the way to fix it is to abandon the LSTM cell and use the regular one, the LSTM.
Okay.
So that's the fix. I think we are going to flexibilize the grading on homework 8 because of this issue. Because I know you guys attempted it and you got numerical instability with the LSTM cell, which is also what I got. And yeah, so we may revise the homework 8 later and let you know what to do next. So the right answer for this would be the surrogate we have for the tellurium model, and then instead of the tellurium model, it would be a parabola, right? That would lead to the correct prediction and extrapolations both to the left and to the right from parameters to time series.
Yeah.
All right. Any other concerns?
Get a face locking model right here in the chat. Just the equation, because I tried to check what I did wrong, but I couldn't get the oscillation with the one which I created from the paper. So
Oscillation? What are you talking about exactly?
from the previous class, um, face locking exercise.
Oh.
Oh.
Are we going back to that, James?
Well, we're basically out of time, but we could take 5 minutes to look at it together. Sure.
Okay. I'll share my screen. Yeah. So I'll keep the paper. So this is the model. I'm trying to keep it side by side, but can you see my screen?
So what happens when you run this?
So I get this as a result.
So I guess, and that means that there's an error in the coding that you got.
But I verified the parameters as well.
So then if there's an error in the parameters, then the error has to be in the way you type the equations.
It could just be that she's not running for long enough.
You could
Maybe
try
it's just-
running it for longer. That would be a good start. What happens if you run it for 10 times longer?
okay.
Yeah. So like fourth, yeah, that's it.
That won't work.
With
Why not? Before you answer, why not? Why won't that work? What does result do? Why will that not work? What is the function of the method result? What does it do?
Mm,
Tell me what the arguments of result are, of simulate are. Sorry, simulate.
So it creates.
No, scroll down.
Okay.
You have a box, a cell called
Yeah, it
results equals,
creates this. Yeah.
right. But what does m dot simulate do?
It will run the model
Tell me
for
what the
this.
arguments of it are.
So this is the starting time, this is the end time, and this is the samples that it create between the start
Right.
time and the end
So
time.
can it possibly, if you're going for 5,000 time steps, can it make any sense that to have 100 steps?
No. Yeah. Oh, the load. Sorry.
I've gotta go, guys.
Okay, thank you, Dylan. We'll talk to you
Okay.
soon. Bye.
I'll email you about meeting sometime this week.
Taking more time than required. Mm.
It wasn't that. So you need to check that you didn't make a mistake when you implemented your equations.
Okay.
So I think that you're going to have to check to see what I don't have the equations. You should always, when you do an exercise like this, in the cell above this, you should copy the equations and paste them in as a figure or as latex so that you have them to compare.
Yeah. I'll do that.
So then that way, if you want, you can screen share, put the equations next to it, and we can look quickly at that together.
Is it not shared right now? Like I thought I'm sharing it with the equation.
No, you're
Oh,
just sharing your notebook.
oh, okay. I'm sorry. So, I thought I'm sharing this screen. I'm so sorry.
It's okay. Very good. Fine. So, ANIN, top. Why is it multiplied by NN in the first one? Where does that come from?
Okay.
Where does it come
That
from?
is wrong, actually. It's wrong.
Why don't you fix it then? Okay, look at the next line. Equation for I is also wrong.
Okay.
Please fix
But
it.
it okay. Um,
No, it's still wrong.
it's KTL into IM alpha.
Help read the equation. Read that line to me completely.
Mm. I decays to at the rate of KTL
Right?
into IM.
Fine. And what does the equation say?
So I is reducing at the rate of
Read the equation in the right.
but
What does the equation in the right in the paper say?
the rate of change of I with respect to T is it is right. Oh, okay.
That's better. What about the other one? See, alpha K, IKK, and Todd. Okay. Yes. Okay. Try running that.
I think I found out. So here IM is reduced to KT into N square, and this is ulta.
Okay. Run that.
Mm, getting EV convergent test failure occurred. Can I ask?
Okay. So then there's another mistake somewhere. Run it for a short amount of time. I mean, at this point in the semester, it's important that you be able to do these things on your own. You should have had enough practice to be able to do this. I'm happy to work with you on it, but it's important that you really work on it on your own, put time into it,
Mm,
because this is fairly basic. And so
okay.
it's
Okay.
easy to make a typo, but it's important that you be aware of the things that you need to work on. Okay, so when you run it for a short amount of time, what happens?
Uh,
Make it 50. No, don't change the number of points. Make it 50. Five is fine. 5 is fine. Okay. So, well, you better do a reset, reload the model before you run it again. Okay, now, okay, so there's a convergence error. That means there usually is a divide by 0.
Mm.
So something is not right. Where okay, Pedro, thank you.
Thank you. See
Uh,
you this week still.
Okay. Yeah.
Professor, maybe I'll take some more time and I'll try to work on this, and then if I still have a problem with it, I can discuss with you
I mean,
at the next call.
the probability, that you can have issues, for example, if you have an uppercase where you expect to be
Mm-hmm.
lowercase, because upper and lowercase are not the same.
Yeah.
So mixing upper and lowercase is always a little risky because it can make things not be what you expect. KI. It's all right. It's okay. Actually, go back to the very beginning, please. The equations.
Okay.
Look at line N1 or R1.
Mm-hmm.
What do you have?
So NN reduced.
What do you have? Read line R1.
Okay. NN reduced to with at the rate of Kn in into n total minus Nn into Ki by Ki plus I.
Right. And what does the equation say?
So the Nn by dt
Yes.
is but
What did you do before? That wasn't
okay.
quite right.
Yeah. Okay.
Can you just, what did you do wrong? There you go. Now try running it.
But it is reduced to
No, you didn't fix it. What did you do wrong? Do you understand what the you know what? I'm trying to understand where your misunderstanding is. I'm trying to be helpful. Do you know what a differential equation is?
yeah, it is the rate of change with respect
Right,
to the
right,
time. Yeah.
right. And look at R1 and look at the equation you have here. Are they the same?
Okay. So this is the reduced term rate, and this is the
Tell me,
product term.
write out for me. I'll use annotation. Write for me the equations that R1 and R2 represent.
Okay. No, I. Okay. So R1 is, once again, R1, so n of n is reduced.
No, write it in equation. What is the equation that represents?
So d of n
Yes, is what?
is equals to k of nn.
No,
So it should be minus because it is reduced, but I have
that's right.
shifted the term. Yeah.
They're backwards.
Yeah.
So fix that. That's the same thing you did in the other line.
Yeah.
Okay.
So the thing is, it was written like this before.
What do you
Like
mean it was written that way before?
like I wrote like that before. Maybe I had that number correction. So I prompted the Gemini and it overcorrected it. So when I tried it
So
out and I didn't
you mustn't use computing to do it. You must do it
uh
yourself.
I'm so
Otherwise, you won't
sorry.
understand it.
Yeah, I am so
It's
sorry
okay.
if
It's
my
okay to use the computing to help you when you understand.
uh-huh.
But we've discussed this. If you don't understand what you're doing, you can't understand when the AI makes a mistake, can you?
Yeah.